Glycolysis is defined as a sequence of reactions converting glucose, or glycogen, to pyruvate or lactate with the production of ATP. Salient features. Glycolysis is a universal pathway in all the living cells. This pathway is often referred to as Embden-Meyerhof-Parnas pathway, EMP pathway, in honor of the biochemists who made a major contribution to the knowledge of glycolysis. Glycolysis takes place in all cells of the body. The enzymes of this pathway are present in the cytosomal fraction of the cell. Glycolysis occurs in the absence of oxygen anaerobic, or in the presence of oxygen aerobic. Lactate is the end product under anaerobic condition. In aerobic conditions, pyruvate is formed, which is then oxidized to carbon dioxide and water. Glycolysis is a major pathway for ATP synthesis in tissues lacking mitochondria, e.g. erythrocytes, cornea, lens, etc. Glycolysis is very essential for brain, which is dependent on glucose for energy. The glucose in brain has to undergo glycolysis before it is oxidized to carbon dioxide and water. Glycolysis anaerobic may be summarized by the net reaction: glucose plus 2 ADP plus 2 pi2 lactate plus 2 ATP. Glycolysis is a central metabolic pathway with many of its intermediates providing branching points to other metabolic pathways. Thus, the intermediates of glycolysis are useful for the synthesis of amino acids and fat. Reversal of glycolysis along with the alternate arrangements at the irreversible steps will result in the synthesis of gluconeogenesis. Reactions of glycolysis. The pathway can be divided into three distinct phases: A. Energy investment phase or priming stage. B. Splitting phase. C. Energy generation phase. A. Energy investment phase, preparatory phase. Glucose is phosphorylated to glucose 6-phosphate by hexokinase or glucokinase, both are isoenzymes. This is an irreversible reaction, dependent on ATP and Mg+2. The enzyme hexokinase is present in almost all the tissues. It catalyzes the phosphorylation of various hexoses, fructose, mannose, etc., has low Km for substrates, about 0.1 m, and is inhibited by glucose 6-phosphate. Glucokinase present in liver, catalyzes the phosphorylation of only glucose, has high Km for glucose 10 m, and is not inhibited by glucose 6-phosphate. Due to high affinity, low Km, glucose is utilized by hexokinase even at low concentration, whereas glucokinase acts only at higher level of glucose after a meal when blood glucose concentration is above 100 mg/dl. Glucose 6-phosphate is impermeable to the cell membrane. It is a central molecule with a variety of metabolic fates: glycolysis, gluconeogenesis and pentose phosphate pathway. Glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate in the presence of the enzyme phosphohexosisomerase and Mg+2. Fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase PFK. This is an irreversible and regulatory step in glycolysis. B. Splitting phase. The 6-carbon fructose 1,6-bisphosphate is split to two 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase fructose 1,6-bisphosphate aldolase. The enzyme phosphotriosisomerase catalyzes the reversible interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Thus, 2 molecules of glyceraldehyde 3-phosphate are obtained from 1 molecule of glucose. C. Energy generation phase, payoff phase. Glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This step is important as it is involved in the formation of NAD plus H plus and a high-energy compound 1,3-bisphosphoglycerate. Iodoacetate and arsenate inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. The enzyme phosphoglycerate kinase acts on 1,3-bisphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. This step is a good example of substrate-level phosphorylation, since ATP is synthesized from the substrate without the involvement of electron transport chain. 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. This is an isomerization reaction. The high-energy compound phosphenol pyruvate is generated from 2-phosphoglycerate by the enzyme enolase. The enzyme requires Mg+2 or Mn+2 and is inhibited by fluoride. The enzyme pyruvate kinase catalyzes the transfer of high-energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of ATP. The end product is pyruvate. This step also is substrate-level phosphorylation and the reaction is irreversible. Thanks for watching. Subscribe the channel for more videos.